All right. I hope everyone can see the slides, can hear me very well, can see my face. If you feel there's a problem, for whatever reason, the internet is not good, my voice is not coming up, please speak up. Okay? All right. So good morning, everyone. Today it will be the real first lecture, although last week we met already for 2 times. We discussed the syllabus, we discussed the grades, the content of the course, the objectives, and we agree on something. Hopefully it will be exciting for all of you. We will have about 50% of the course dedicated for the review paper, and the other 50% for the programming assignments, where we will try to build a read mapper over the course. We will take it gradually, step by step, and start building each of those steps while we explain them in the lecture. We go over the material, we discuss how it works, and then your job is to implement those tasks and show that it's working. And by the end of the semester, hopefully you will get a full implementation of a read mapper, which is a key step in all bioinformatics application. And it's hard to see a bioinformatics application that does not require comparing few texts or few sequences to each other. Today, it will be more as introduction to bioinformatics. So hopefully most of you are already aware about some of the contents, especially those who studied biology during high school or during bachelor degree and so on. And some of you are already working in bioinformatics topics, so that will be something interesting to build up a strong foundation and then move on. All right, so unfortunately, this week and the last week was a winter storm, so it's still happening in Georgia and Atlanta in specific. So please stay safe and warm, and hopefully by next week we will meet in person and taking all the lectures in person, which will be more exciting than online. Okay, so a brief introduction. I'm Mohammed Alser. You can call me Mohammed. Alser means actually secret in Arabic. I'm working as assistant professor in Georgia State University. This is my 2nd month here in Atlanta. I was in University of Southern California before I came here. And before that, I was for about 5 years at ETH Zurich as a lecturer. And I spent some time in University of California, Los Angeles. I was working in computer science department with Zahar Iskan, where we were working on metagenomic profiling, something related to bioinformatics. We will discuss that in one of the lectures. And I spent some time in TU Dresden during my PhD to do some internship. And I was working before doing the PhD at Petronas, which is a petroleum company in Malaysia. I was analyzing some data coming from the mid-ocean data. I did my PhD in computer engineering at Bill Kent University, graduated in 2018. And during that time, I received the Best PhD Dissertation Award from IEEE Turkey. I finished my master's in electrical and electronic engineering from University Technology Petronas in Malaysia. And I did my bachelor in computer engineering in Islamic University of Gaza in Palestine. I'm currently working in bioinformatics, computational genomics, metagenomics, which is very much related to the topics we will discuss in this course. And I'm currently leading Alser Lab, which is a new research lab at Georgia State University. We have openings for PhD and postdoc. Hopefully, we will have more openings in the upcoming days. We start strong collaboration with the industry. So we'll have more exciting projects in the upcoming month. So please follow up and stay connected with our website to check for more updates, more openings, and to have an idea about our exciting research. I'm a bit active in social media, although it's been very busy days those 2 months due to the movement from California to Georgia State. But yeah, I'm a bit more active on Twitter. I try to post many things about research, about bioinformatics in general. The best way to reach me is basically the Georgia State University email, but feel free to reach out to the Gmail as well. My office currently is in 25 Park Place building, the office at the 18th floor. So 1823, this is the office number. So feel free to visit me or discuss over email, and then we can arrange for a time to meet or during office hours. I'm excited to have strong TA team. So I have Lakshmi, who will join us within the upcoming days. He will be helping in grading and helping in the assignments whenever it's needed. Okay, so I have one question for everyone currently online or on Zoom. Have you ever heard about bioinformatics before? Let me see a few hands. Please raise your hand if you hear about it. I would like to see more hands. All right, I see four persons. Okay. Would you mind
hearing from you? Does
yes,
hearing
please.
From you counts or no?
Sorry?
Excuse me.
Professor, does hearing from you counts or not?
You cannot hear me well?
I can hear you. Can you hear me?
Yes, yes.
Does hearing from you, the bioinformatics, hearing from you counts or not?
I see. Yeah, let's say so. Yeah.
Okay.
All right. So any one of you can help us, tell us where exactly did you hear about bioinformatics, whether it was a course in the past, whether it was research seminar, whether it was YouTube video. Any volunteer?
Hey,
Yeah, go ahead.
I have been taking classes like introduction of bioinformatics, so I heard from there, and I attended few conferences in Georgia State University as well. So yeah,
That's amazing. Yeah, who's next?
Yeah. So to be honest, I Googled it and I found this course is like fascinating. And
You Google it or Chat GPT it?
no, I Googled it. Yeah, I Googled it and I found this course is very interesting. And yeah, then yeah, then I registered. Yeah.
Indeed, yeah, it is really interesting to see how really computer scientists or anyone basically from any field, biologists, chemists, how they can really help in this field. So we'll see more exciting example in this course, and I have really nice applications that would benefit from interdisciplinary field such as, again, math, biology, chemistry, computer science, electrical engineering. All of them can contribute to bioinformatics. All right. So basically, when we talk about bioinformatics, let's really know the definition so that we can have a strong knowledge about it. I try to ask ChatGPT to build an image that represents bioinformatics. And what we see in this picture, this is again coming from ChatGPT. And you'll have a DNA, as you can see over here. We have some cells. Probably this is maybe some atoms, some chemical reaction happening in the background. We have maybe some statistical analysis going on to the left side. We have some people wearing the lab coat. We have some computer We have maybe some digital data in the floor. What else we can observe? Something that is really wrong, that all the people wearing lab coat. So this probably could be a misunderstanding of the field of bioinformatics, that most of the people working on this field, either they are from medical field, biomedical, or maybe wet lab, where they do the interaction, all those chemical molecules during the lab time. That is something I would believe is wrong. It's part of it, yes, but it's not everything about it. I ask again for another picture, and this is what I got. Again, we have DNA, we have some chemical atoms over there, we have another person in lab coat, we have a computer, we have some tools from the lab, and everything is related to wet lab again, which is another misunderstanding of the field. So we have to go to the formal definition of it, and the best place to get that is the National Human Genome Research Institute. They define bioinformatics as a scientific interdisciplinary, uses computer technology to collect, store, analyze, and disseminate biological data and information. So those biological data, it's not only a DNA, it's not a genome, but there are different forms of those biological data. It includes, of course, the DNA, amino acid sequences or protein, annotation about those sequences, and many more. You can think about database, you can think about algorithm, you can think about program that do something, you can think about mathematical equation as statistical analysis, and so on. So it's really interdisciplinary, and we don't say multidisciplinary because it's not like disconnected field, but rather many fields that work together. Without having some strong understanding from biologists, we as a computer scientist, we cannot do anything in this field. So we have to understand from biologists how it works, and for them, they have to understand how algorithm works so they can use our tools that we develop in their problem. So it's basically really interdisciplinary field where all the people coming from different knowledge, different background, work together to solve some problem in biology and some problem in medicine. When we look at the market, the worldwide, basically, not in a single country, it's expected to reach about $100 billion in about 2028, which is really not far from 2025. And this number is huge. If you want to get more imagination about how huge this number is, look at the market for cloud computing, for example. So think about Amazon Cloud, Google Cloud, Microsoft, all of them together combined have $100 billion of market share. It's similar to video games or artificial intelligence market, which is recently evolving rapidly, including NVIDIA, Google, IBM, and so on. So this number is huge just for bioinformatics field. And we will look at the amount of data that is generated from many sources. We will find like four major sources of big data, including astronomy, Twitter or X, genomics, and YouTube. And when we check the governments around the world, each government has its own genomic projects that try to understand the genome of the people living in that country. And that helps them to identify potential diseases so they can develop drugs or build plans for hospitals to have some precautions before the pandemic happening or some disease starts spreading in the country. And it's not only about the human, but also about vegetables, fruits, all the crops. So basically, you need to protect them from potential disease. And we start seeing the importance of fields such as bioinformatics after the COVID-19 pandemic. So when it impacted the entire world, most of the governments start dedicating most of the research grants toward something related to the biomedical field or COVID-19 in specific, or many other virus detection or research on genomics. All right, so hopefully this gives you a formal definition of bioinformatics, but what is actually a genome? When we look at this picture, we will see something interesting. We will see people of different faces, basically, and this is the norm in most of the countries. But when we start understanding what is happening, why those people have different skin color, why those people having different hair color, body mass, length, weight, and so on, is it really a disease? All of those people could be healthy, right? But we still have differences. So the first question, what makes us different? Second question, what causes a disease, right? Why we have disease, basically. And why does a disease or a medication affect us differently? So we might have two person with a disease, right? And we get the prescription for certain medication, but one of them getting better after the medication, but the other person is not getting any better. Why is that? And when we thoroughly and deeply try to check what is causing those differences, it's basically our genome. So the genome is really organism complete set of genetic instructions. And when we talk about instructions, it's coming from computer science. Instruction is something that you are telling the others to do something, right? So this genome is really telling the cell itself how to act, how to live, basically. Whether it should be healthy one, whether it should have some disease, which should produce some protein that causes something to observe on our body or not. And it's really, again, not about a human. It's about viruses, bacteria, plants, fruits, vegetables, and even some animals. And out of our curiosity as a human being, we start even checking on the other planet, whether we have some livings over there or not. So this genome or genomic material exists in most of the living around us, which is really important to start studying those genetic material or genomes, basically. And when we check the content of genome is basically a text. So that is the simplest format that we can think about. So forget about chemistry. Now we can convert everything in our genome as a text format. And when we check the chemistry behind it, we find it only 4 molecules. Think about how really great this genome can do everything to us, can impact us negatively or positively, but at the end is just 4 chemical molecules, which is adenine, guanine, which is we denote as G, cytosine, which is C, and thymine, which is T. That's why in this text format, you can see that everything is just enumeration or combination of those 4 characters. If we replace any of those characters with something else, it might lead to a disease, or it might lead to something that is not a disease, but a different phenotype. For example, eye color is not a disease, but it's just something different that we can observe from the outside. But it also can be inherited to the second generation. So it can go with the DNA to the children, right? And then it might cause a disease to them, but not to us. All right, so we would like to understand how large such a genome. How many of you know this building? Let's see some faces. We have 1, 2, 3, 4. Please raise your hand if you know this building. 5, 6, 7. All right, can any one of you tell us what is it? Quickly, who's first?
That's 55 Park Place, right?
Excellent. This is 25 Park Place building, where we have the computer science department. And I would like to compare the human genome with that building. So if we try to print the human genome on letter pages and we stack them on top of each other and build a tower out of those paper, how large this tower could be compared to 25 Park Place? Is it taller, shorter, same length, maybe way shorter? Imagine how crazy is this? Just printing the human genome with a small font size, no spaces between the line, very little margin around the page, and then you stack as many pages as you could on top of each other. Any idea? Yes, Vemeth would like to tell us. Is it shorter, longer? Okay, let's see. It's a bit longer than this tower. Imagine how crazy is this. So that is a human genome in a single cell in our body. Single cell, not in the entire body, just single cell. In ours, we have about number of chromosomes that build our genome. A single genome printed on those pages forms this huge tower. And when we try to compare that to the other species, for example, this virus, the Phi X174 virus, we see the genome size about 5,000 base pair, 5,000 character. In the previous slide, it was about 3 billion. Over here, human genome is about 3.2 billion characters. In the viruses is normally in a range of thousands. When we check bacteria, for example, the famous E. coli is about 5 million character. And by the way, this virus can infect this bacteria, can cause a disease to this bacteria. When we check a human genome, again, it's in order of billions. However, we are not the best in terms of the genome length. We have red onion, for example. It has 5x longer genome than human. And in the past few years, just before like 2 years, the largest genome we could read until now, or until just 2 years ago, it was this Japanese flower called Paris japonica. It's about 50x longer genome than human body. However, in the last year, we are able to read a genome of an animal that is believed to be the largest ever we could sequence. And this called the South American lungfish. In 2024, there's a research paper that are able to decode the genome of this living, basically. And it's open source. So if you're interested in seeing the content of that living, so I would advise everyone to go through this link from NIH, National Institutes of Health, and try to download the assembly, check the file size, check the content, read what is inside it, and so on. It's open source, it's public, it's for free. You can go ahead and download it right away. All right, so now we would like to have a closer look into our genomes as a human being. So when we check any cell in our body, you will see that the genome is basically not a single sequence or not a single text or paragraph, basically. It's basically divided into 22 pairs of chromosome plus the 23rd chromosome that represent the sex, male or female, basically. And each of those 22 chromosome pairs is actually, for example, if you look at chromosome 4, we will see that we have 2 chromosomes. That's why we call it pair, right? And this chromosome coming from the mother side and the other one coming from the father. So you got from the father, from the mother, and then you form those chromosome pair. That's why whenever you have a disease, or we call it a mutation, or changes in the DNA in the parent, it will be inherited to the children, basically. But not necessarily they will be infected with that. Their DNA might hold those changes in the DNA, but they might not get the disease ever in their life. They might also pass it to their generation. So we'll get one chromosome from mother, one chromosome from father, and we will look in depth to that single chromosome, not to the pair, but single chromosome. You will see its double helix structure. We have a DNA coiled around each other. So you have one strand, we call it, and the other strand coiled around each other and has some chemical bonds such as it's still connected to each other. And when we check carefully to those chemical molecules, we will see that we have those 4 chemical molecules within that DNA or within that chromosome strand. So we have the adenine, which we represented as A, and then thymine as T, cytosine C, guanine G. So we have A, T, C, G. And normally the A will be chemically bound with the T, and the C will be chemically reacted with the G, and vice versa, G with C and T with A. So based on the color, you can decode those structure. All right? And this is a real picture of those chromosomes. So this is not something artificial. This is not something out of our imagination. It's coming really from the cell of our body. This is a single cell. You will see those chromosome pair. And in fact, one of the chromosome pair represented here, you can see that this is single chromosome and this is the other chromosome. It's, again, it's not really well arranged single sequence, but rather it's super coiled around each other. That's why a single cell has 3.2 billion chemical molecules, and still you cannot see it with naked eye. It's really small single cell because the structure of those chromosomes is really super coiled. It's taking very few space. It's not that it's huge. It's not like unfolded such that it will take very long space. And one interesting thing about this picture, it's coming from a person called Henrietta Lacks. How many of you heard about Henrietta Lacks or HeLa? HeLa or Henrietta Lacks? All right. So this is the lady. This is the lady, the actual lady that owns those chromosomes. And this is an actress that participated in a movie about this story. It's a really interesting movie. And I would recommend everyone to watch that movie. So the movie called The Immortal Life of Henrietta Lacks. And let's watch this together. This is the trailer for the movie. It will be very interesting.
It seemed like a dream. About our mother, could this be true? You don't understand this? We didn't know nothing about nothing.
Scientists have been trying to get cells to grow outside of the human body, but they would always die. Until Henrietta's cells came along.
I want to write a book about your mother.
Go healer, go healer. That's my mother. Hope I don't regret this.
Everyone's saying Henrietta Lacks donated themselves. They didn't donate nothing. They took them and didn't ask.
Still is guilty. Somebody's going to pay.
God wanna have a disease cure, he provides one for himself.
I'm not doing anything behind your back. Henrietta helped develop the AIDS cocktail, chemotherapy treatments.
Not to get caught up in hope when you've been powerless for so long. I know I'm a part of you. Can you pardon me?
Right? So to give you some context about this movie, or about this real story. This person called Henrietta Lacks. She suffered from cancer in the past, and then she went to a hospital called Johns Hopkins. I think all of you are aware about this hospital. This is a very famous hospital, very active in research in cancer. And they were very curious to study those cells coming from this person, which we call it HeLa. And they took those cells, they protect them, they keep them in nitrogen, liquid nitrogen, for a very long time to study those cells. And even they were able to replicate those cells to make them much more. So they grow them in the lab and they replicate them, and then they distribute those cells to many research centers around the world. So they become one of the most famous cells in the world to study genomics, basically. And they did all of this without her consent. So they took the cells, they replicate them, they distribute them to the entire world without taking any consent from her side. Later on, this creates a lot of problems. It's not about cancer only, but rather it's about the genomic material inside the cell. So people that were aware about the content of the genome, and then they were aware about the content of the genome of her relatives, not only her person, but also her children, relative, uncles, and so on. Why is that? Again, the genomic material is something inherited from parent, and the genomic material of the parents is also inherited from the other family members. So if you are able to decode the content of one of the members, then you will be able to guess the content of the brother, the sister, the other children, and even the parent, which is something crazy because you're going to know about potential disease that those member could have. And then later on, some health insurance companies could block them from getting the policy for next year because I know that those person will have also maybe cancer or something like that. Then it will cost me a lot of money. So I'm going to block them from that. So you can think about potential problems by sharing the genomic material. However, don't worry about that because hospitals are still doing DNA testing, but these days it's much more organized than in the past. So there are consent, there are some control on what to share exactly, what portion of the DNA to be shared, what portion of the DNA to be studied for the corresponding disease that you are going to the hospital for, rather than sharing the entire content of the DNA. So I would recommend everyone to watch that movie to get some sense about this content. All right. So as you are aware from biology class that there is what is called a central dogma of molecular biology, where we have the cell in our body has a DNA, and this DNA has all the what we call as instructions, the things that we were explaining in the beginning of the class. So those instructions, or as a DNA, some of them might be active, some of them might be deactive, which we call RNA. So the DNA will be converted to transcripted into RNA, and then the RNA will be converted or translated into protein. And those codes or instructions will be in DNA format, but what we observe is the protein side. We don't observe the DNA, but we observe only the protein, which is the thing that we call phenotype. So this phenotype is the skin color, hair color, eye color, all caused by proteins. You can think about it as a cookbook, and this book has a lot of recipes, and then one of the recipe will be the RNA, the things that get converted into the food. And the food is the protein itself, is the thing that we observe, may taste, and so on. All right. When we look at our body as a human being or any other living, so you will see that our body forms of cells, and each of those cells has the genome inside it, right? But what makes a cell of the heart, for example, different from the lung, different from the eye, different from the tongue, the skin, and so on, although all of them have the exact same genome, it's not different from one cell to another. What is different is how those instructions will be enabled or disabled. So you can think about it as very large number of switches, and each of those switches could be on or off. And this is how the cell will be different from the lung to the heart, hand, tongue, skin, and so on. But all of them will have the exact same genome or DNA, except the cancer. The cancer is where the genome will get affected by, for example, some radiation or some other factors that affect the DNA itself and get it to be changed. So think about a heart cell is appearing in the lung. That is a cancer directly, because the genome is not supposed to be there of the heart in the lung, right? So the DNA itself, if it's get changed, then that is causing a cancer. All right, hopefully now we understand what is bioinformatics and what is a genome. And now it's important to understand or read that genome. But how we do that? How we go to the cell and extract the genomic material and start reading the content or getting the full text out of it. So one of the first steps you need to do is basically to do the DNA testing. And this testing you could do yourself, or you could go simply to the hospital and ask them for DNA. Hopefully the health insurance will cover that. But if you'd like to do it yourself, you go and purchase one of those online kits for DNA testing, for example, 23andMe. And then you pick that swab, you take it from any material in your body, and then you send that tube to the company back. And then the company will do something for that tube. So the cost currently is about $1,000 if you want to do the full comprehensive test, for example, online. This is not advertisement to them, but this is just an example how you can do it. There are many other companies, many other services that you can do. And once you send that tube to the company to read your material, we expect them to send you a report of the 3.2 billion character of your body, or if you're that DNA. However, until today, we have no single technology, no single machine that gives you the full content of your genome. That's amazing. Why is that? If you are really curious, I would recommend you to read that section 2.1 in our paper that build on top of the whole story. However, what we got from the machine, again, is not the full sequence of your genome. What we got is basically the lab technician, or in the wet lab, will do something to the material to isolate everything else from the cell, because we have plasma, because we have white blood cells, red blood cells, and so on. It will isolate all of those and keep only the nucleus of the cell, what is inside the cell as a DNA. And then try to shred that DNA, because again, it's super coiled around each other, and we will be unable to read that content if we keep it as is. So we'd like to break it down into smaller pieces so we can read each of those pieces individually. And they do that by rotating the tube at very high speed, or apply ultrasonic radiation to the sample to break down that DNA. And then they send those pieces to a machine that we call it a sequencer. This is a machine, a physical machine, that you send the DNA or the shredded sequences to that machine. The machine will give you a picture or a movie or even electrical signal and amplitude. And then our job is to convert those material that we got from the sequencer into what we call genomic sequences or a text format. And we do everything after those pictures, the electrical signal, or the movie, or the video, basically. We do all of that in what we call a dry lab. So everything done at the bottom is done in the dry lab or computationally. Everything at the top is done in a wet lab, physically. So you have to have some chemical molecules added. You have to do some mechanical job. You have to send it to the sequencing machine, which also involves some chemicals inside it, and so on. In the next lecture, we're going to visit in depth the sequencing machine, how it works, what kind of chemical reaction happening inside, how we basically convert the chemistry or the chemical molecules from this format into that format. So basically, as mathematician or computer scientist or any other field, most of them will contribute to this thing in the bottom. However, if you are from chemistry, for example, you might be developing something related to those chemical reactions. But still, you might be able to contribute something over here, for example, understanding the structure of the protein itself. Currently, we have 3 major companies that develop those sequencing machine. Again, next lecture, hopefully we're going to visit each of them in depth and understand how it works. But to give you an idea, this is a benchtop size called nanopore. It can accommodate up to 48 samples at the same time. So you can put 48 genomes and try to read them, all of them in parallel. This is a fridge size. It's not a benchtop. This is only the benchtop machine that we have. This is still fridge size, a huge machine. This is another huge machine from Illumina. And those coming from different companies, although now recently PacBio start to shift to either this technology, or basically something similar with having smaller devices and producing short sequences or wide range of sequences. You can see that they are different in the read length. For example, this is very short sequences, but very accurate. The moment you move from short to long sequences, you start losing something in the accuracy, and this is expected. Now, none of those machines, again, give you the full sequence of your genome. What they give you, pieces. As you remember when we break down the genome into smaller pieces in the chemistry format, and then we start to convert those sequences into text format, those are the pieces that we are getting actually from the sequencing machine. And our goal, think about it as a puzzle. So we want to solve that puzzle by having the reference picture and having those individual pieces. And you start picking one piece, try to compare it with the picture, and then identify the potential location. You might make some wrong guesses. That's fine. You can correct them later on as you move on, as you build the puzzle, right? But it will be very hard if we think about those puzzle pieces as very small. Think about large quantity puzzle, like 1,000, 2,000, 10,000 pieces. This will be much harder, right? However, it can be very hard if we don't have the reference picture, if we don't know what we are solving. So you have all those pieces individually, and they're not giving you the reference picture, right? So you need to guess. You pick one piece, another piece, try to compare whether they have the exact same color, like blue, then potentially those might be next to each other, representing the sky, for example. Or you might get 2 puzzles, and none of them has the reference picture, but you know that those 2 puzzles are exactly the same thing or representing the same individual. And this is exactly what is happening in the cancerous cells. So we pick one cell from the same individual, assuming it's healthy, and we pick another cell from the cancerous cell itself. And we have healthy cell and unhealthy cell, and we try to compare the genome together. We don't have the reference picture because we don't know the reference genome, because we didn't build it in advance, but rather we're getting from the 2 cells. And both of them, we do the sequencing step, which is breaking down into smaller pieces, and then send it to the sequencer, and then start reading those pieces. So this is a very difficult problem. However, the problem can be much more difficult if, again, we don't know what we are sequencing. Think about that we take a swab from the classroom seat. So we don't know what is in there. We don't know if it is fruit, vegetables, if it is a human being, if it is other organism, right? And to solve that, as if you are solving the puzzle, you need the reference picture. But we don't know which reference picture to pick. Think about many puzzles, you scramble them together. And then you don't know which is for which reference picture. So the only way to solve it is to bring all those pieces, try to compare them to the old reference pictures that you have in hand, and try to guess whether this color does not match this picture, so let's put it aside. And then you have a shorter list of those reference picture, and then you try to do fine-grained checking for all those pieces. So this is still a very challenging problem. And there are many other examples where you can see that this solving this puzzle, it's still a very tough problem with the current technology. However, if the sequencing technology got updated for whatever reason, some guy developed novel technology that is able to read your genome as a whole, as the entire complete sequence, that will do a revolution in this field. And I believe we'll get Nobel Prize for sure on that. But until today, we don't have such technology. All right, so now we already know how to read the genome, hopefully. We will go in depth again for those sequencing machine, how it works, how we generate those pieces in the next lecture. But now, why to read? Why to analyze the genome? Is it only about disease? Is it really we need to know those diseases and solve them? Let's see. So there is a famous saying by Richard Hamming, who's a famous American mathematician, says, the purpose of computing is to gain insight, not numbers. So whenever you would like to analyze something or do some thorough, in-depth analysis to that thing, you're not really worried about the numbers or the raw data, basically. You want to get more insights, like how it works. What does this portion of data represent, right? Rather than just checking ACG, TTCG. It doesn't give you much of information. But when you know that this location in the genome is responsible for this kind of thing, then this is becoming more interesting. And this is exactly what we would like to have in bioinformatics. We need to gain insights and observations much more efficiently than ever before. Why is that? Because now the scale is different. Now we are not studying a single human being, but rather we are studying thousands of human beings, maybe a million. In the COVID-19 time, before any single flight, we were testing every single passenger in the airplane. So think about the scale. This is grand, really large, grand scale that we are trying to analyze. One of the most famous application that we are studying is basically we have several individuals and we read the DNA of all those individuals, assuming that we solved the puzzle already and we got the full picture about the genome of each of those individuals. And we have a certain disease. Let's call it, for example, here, blood pressure, right? So when we take the reading of those blood pressure for each of those individuals, we have here about 16 individuals. We have different readings for the blood pressure. We see two groups, basically. Or maybe we can divide them into three groups. Low blood pressure, somehow mid-range blood pressure, and very high blood pressure. When we check the genomes of those 16 individuals, we see somehow it's exactly the same. You can see most of the characters are exactly the same, except two variations. We call them SNP, SNP, single nucleotide polymorphism. That is a SNP, or single variation. When we check those single variation, the two of them in depth, we will see that we have SNP1 and SNP2. Now, our goal is to identify which one of them is responsible for high blood pressure. I'm sure all of you are smart. So can you tell me whether it's SNP1 or SNP2? Yeah, please enable your microphone. We have 16 individuals over here. We have the readings of blood pressure. When we compare the genomes of those individuals, we will see that everything is exactly the same, except two columns.
Is it SNP1?
Excellent, excellent. That's great. SNP1. Why is that?
Because it's all C as the blood pressure begins to increase.
Exactly. That's great. Excellent. So when we see the low blood pressure, we will see we have in SNP2 is really random. AEG does not correlate with what we have in the reading. But when we check SNP1, we will see that C's always correlate with high blood pressure. right? But when we have T's over there, it's always correlate with low blood pressure. That is telling us whenever you have C in that particular location, then we expect that person to have high blood pressure. Again, this is not necessarily to be currently having high blood pressure. It might show up as you grow, or it might just be inherited to your children, basically. So any genomic or any genetic variation, it might not represent a disease currently, but you might holding it, you might having it in your genetic material, and you might pass it to the next generation. And this is a very famous study, and we call whatever you saw in the previous slide as genome-wide association study, or GWAS in short. What we do over here, we take normally two groups of people. One group that we call it a control. Those are people without having that particular disease that we are studying. And another group of people that we call it a case. And those people with that particular disease that we are studying. And our goal is to identify in a population what might cause that disease as a genetic material. Why we are taking a large number of people rather than comparing a single healthy with single unhealthy person because of the accuracy. Some of the disease might not be caused by a single variation. It might be multiple SNPs that all together contribute to that disease. So it's really not an easy job to identify that single change in the DNA. And also due to the nature of those genomes. So the genome might represent many other diseases, although we are studying single disease, but we cannot isolate those locations that are responsible for that single disease. So when we study genomes, we are studying already multiple diseases at the same time. That's why we are collecting a large number of people, hoping that by having many other cases, we are increasing the accuracy of those detection. And then we build what we call a Manhattan plot out of all those genomes. This Manhattan plot, you can probably infer why we call it that. You can see all those high towers. And this is the x-axis where we have the chromosomes. We have chromosome 1, 2, 3, 4, and every single location in those chromosomes. And then we do statistical analysis for how many times those variation appear in the unhealthy individual compared to the healthy individual. Whenever we got high frequency of those variation, that is an indication that all those SNPs might be correlated to that disease. And then we do thorough analysis to identify which of those SNPs that have high variation or high frequency is responsible for that disease. Currently, we start studying not only the DNA, but also the other genetic materials such as RNA, protein, and do similar studies to the GWAS, but we call it TWAS, where we have transcriptome-wide association study. So if you're interested in this field, you can refer to this paper and check it in detail. However, hopefully the previous part is not boring. I always, as a computer scientist, like to do the things practically. So practically, how we do it, how we check those variation and check the correlation between them. So there is an open source database called opensnp.org. Everyone can open it. It's for free. It's open source. When you go to the top menu, you will see data. And when you open it, you will see SNPs. So if you click on SNPs, you'll get something like that. You'll get first a list of those SNPs, and then when you click in any one of them, for example, I choose this SNP, you'll get this page. What this page tells you is that this is the name of the variation or the SNP, the single nucleotide polymorphism, and this is the chromosome where it appears on chromosome 19, for example, at the position this, and the weight of evidence, we have about 900 individuals having that SNP in their genome. Now, what is more interesting is to see what that SNP causes, right? So, do you remember the story I was telling you about the chromosome coming from the mother and chromosome coming from the father? That's it. So if we have a T from the chromosome coming from the father or the mother, and another T from the other chromosome on the same chromosome pair 19, then we have about 20 to 25%, right, of such hepatitis C patient respond to treatment. So we have very low probability that patient will get better with the medication. However, if they have C and C coming from the father and the mother, then 80% of those patients will probably get better. So you can think about that we might know the result of the treatment even before trying. Rather than letting the patient to suffer, we prescribe some medication for them. We let them try for a few months until their case gets worse, right? And then we say, oh, maybe the treatment won't work. Let's try something else. So this will save time, will save health, will save a lot of money by not try and error with the patient, but rather checking a personalized medicine. So whatever you have in genome, we prescribe medication based on that. And that will be the right way to do it. Out of curiosity, you can go to OpenSnap, check any other snaps, and see the exact same example or maybe other examples for other disease, and see how really those snaps will affect our health and our treatment plan. Again, those snaps might not be as simple as single variation in our genome. Could be something we call structural variation. Structural variation means a variation that is longer than 50 character, or a change that is longer than 50 character in our genome. So it could be substitution, could be insertion, could be deletion. Think about a piece is deleted from the DNA due to whatever reason is. So let's see some examples. So all of those 4 diseases, they are affected or caused by a variation in chromosome 16. Chromosome 16. However, if we have a deletion in that chromosome 16, check the amount of the deletion. It's a huge. 500,000 characters get deleted from chromosome 16, and that probably causing the autism, right? When we check the obesity, for example, it's again deletion of 500,000 characters. Those characters different from those characters. And this is a real study. So you can see here the reference of that study. In the second line, if you copy it and paste it in Google, you'll get the paper that discovers that. However, if you see the opposite disease, like obesity and underweight, you will see the totally opposite effect. Instead of deletion, we have a duplication of 500,000 characters in the same chromosome. So it's amazing how those very large variation is causing a single disease. It's very hard to detect such variation. And there are a lot of efforts since more than 30 years, and we're still far away from having a systematic approach to detect those variations. There is a very nice survey paper about structural variation. I would recommend everyone to check if you're interested in this area. Rather than having OpenSnap only, we could have another database that represents all those structural variation. Currently, I'm not aware of any database that gives you those structural variation. We have it as individual disconnected papers. Maybe there is a database somewhere, but I'm not aware of it currently. All right. So building on top of the story of those variation and OpenSnap, there is currently in the UK, there is a huge effort to do what we call personalized medicine. And there are a lot of studies showing the feasibility of such analysis, to do genomic analysis before prescribing a treatment to the patient. So most of those studies agree on that performing such whole genome sequencing for elephants, those critically ill elephants, once they get born, they might have some genetic disease. And if we treat them, or if we test their genome before we treat them, we will directly avoid morbidity, reduces hospital stay length by an important fraction, and reduces inpatient cost by an important amount of money. That's why in the UK, starting from 2019, every single seriously ill child in UK, they directly offer them whole genome sequencing as part of their care. Hopefully that will save cost, time, and also life. And this is coming real. This is currently in most of the government trying to do that. So this is an important paper published in 2019 in Nature Communications. They try to build a map for understanding the genomic variation of about 50,000 Icelanders. However, the computational cost to do that is huge. Think about 4 million CPU hours, or 83 CPU hour per sample, just to check the variation of 50,000 Icelanders, not the entire population, right? Think about America, for example. If you want to do that for the entire population, that's a huge workload to be done. We start also seeing a very large need for those genomic analyses to understand the content of the virus that causing COVID-19, or in this example, Ebola. However, most of those, like pandemic caused by bacteria or viruses, we still don't have strong understanding of what is their genomic material, for a simple reason that over time they are changing their genomes. The genome itself getting mutation over time. So every 3-4 months, we start seeing different or new mutation in those genomes. So it's not like, okay, I want to understand now the genome of the COVID-19 virus, and I understand it, I sequence everything, and I store it somewhere and done. No, it's not like that, because every few months, this genome will get changed. That's why it can make the vaccination ineffective after some time. So every once in a while, we need to repeat that process, start understanding what is happening as mutation. This mutation might coming from different environments, like different country visiting the US, for example, and those patients holding that virus, bringing in a new mutation to the country. Or those mutation could be developed inside the host, which is the body, ourselves, right? So over time, the virus might get stronger and stronger and then change itself to tackle or to trick the immune system that we have in our body. There is an important study done by UCLA on how we can use genomic testing, DNA testing, to detect the COVID-19. The time needed, it's not as fast as those quick testing that we do for about 3-4 minutes or 15 minutes. Those kits, like we could get commercial kits from supermarket, try to do the testing ourselves. Of course, this is taking longer time because there's sequencing involved. However, the scale could be large, huge. Like we could analyze 1,000 samples in the same time rather than one by one. And this is very important for public health. Think about crowded places as in downtowns, for example, or New York subways and so on. So we have large number of visitors, tourists, and many other people. If there is a disease spreading over there, it will be very great, nice to understand what disease we have and try to do precaution before it starts spreading to the entire population. And what we call that, we call it as metagenomic profiling. Meta means fake, genomic, or a collection, large collection, either fake or large collection of genomes. So we have metagenomic profiling, meaning that we try to understand a collection of genomes in a population that we don't know, we are not aware of. So we're trying to understand what organisms are present in a given environment and how abundant are they. There are a large number of studies doing that. This is one of them, published in Nature 2022. Try to build and profile about 3,500 genomic assemblies that is requiring about 28,000 virtual Amazon Cloud CPUs. Again, this is a huge workload. That's why there are a lot of work on optimizing those algorithms, building efficient software. It's not about just implementing Python script, but rather requiring really questioning every single line in the code. Why we need that? Can we make it more efficient? Can we replace it by more efficient library and so on? There is also a study in New York subway try to analyze most of the station in the city. So they collect about 1,500 samples from those stations, and then they record the GPS coordinates of those samples or those locations, and try to build a map out of it and see what is the dirtiest place in those stations, or what is the most threatening location in those stations. And then what they found is really interesting. About 50% of the things in those samples is about known bacteria. We already know those bacteria. It's not necessarily to be harmful. Sometimes bacteria is really useful. It can digest, or it can really attack other living, so to make the life balanced. But there are about other 50% unknown organisms. We don't know what they are. We don't know whether they are harmful, whether they are useful, whether they are bacteria, virus, and so on, which is really interesting. So we're still far away from understanding what is happening on Earth. This is another more recent study. As a follow-up on this study was in 2015. The most recent study in 2021 tried to build a map not only for New York City, but rather for 60 cities around the world. And they collect about 4,700 samples from different stations, from different classrooms, from different locations around the world, and try to build this map. This is online, already available. You can visit it. You can check the map of the things living around us in the world and see what is happening over there. This is, by the way, part of initiative called MetaSUP. So you could participate yourself. They could send you a kit. You can collect a few swabs and then send them back to the company. They do the sequencing, or to the basically consortium. They will do the sequencing. And then you can participate in that, in analyzing the data and so on. Who's leading that effort is Christopher Mason. He was visiting Georgia State University as part of the conference we have, IC CAPS, last week. And there is something interesting in the previous study, in the 2015 study. Remember, this study was in New York City. What they find, that they found traces of Blake, which is Yersinia pestis. And for those doesn't know, this Yersinia pestis bacteria is actually what caused the epidemic in the 14th century that killed more than 1/3 of the population of Europe. So think about how crazy to find such bacteria in a crowded place such as New York subway. And this was on news for a bit of time. They were thinking of closing all those stations that's impacted by this bacteria and try to clean it and do thorough analysis and see what's happening there, where it's coming from. However, when they do that analysis, they didn't find any trace of Yersinia pestis. And then they start calling it as a failure of bioinformatics. Failure of bioinformatics. Why is that? Because if you remember what they were doing in 2015, they were using some of the bioinformatics tools to analyze what is happening in the sample. And they could do some mistakes, right? Because they are comparing something that they don't know what is it with everything that they know. And maybe by chance, they get some matches between their sequencing or between the pieces of the puzzle with some reference picture. That is happening because we're just looking for matches, regardless if it makes sense or it doesn't make sense. And by some chance, we get a hit, we got a match between those. And then this is another concern about those bioinformatics tools, is not about getting the tool to work, but rather to getting high accuracy out of the tool. We are participating, me, myself, and many other professors from Georgia State University, in the initiative called CAMI Challenge, Critical Assessment of Metagenomic Interpretation. This is a worldwide initiative where we do analysis for all the existing tools that try to solve that problem, which is metagenomic profiling. And then we are telling or informing the people to use those tools or to not use the other tools based on the thorough assessment that we are doing for those tools, to tell them this is suffering from accuracy, this is suffering from large execution time, this is suffering from large memory, and so on. And this challenge is open to public, so anyone can participate. They normally give you the data, give you what they are looking for, what is the end results, and your goal is to develop that tool that can identify what exists in any sample. And based on the accuracy, based on some challenges, they can identify the winner out of that challenge. We have many other papers in this topic. So if you're interested in this, or if you pick a review paper on this topic, feel free to check this paper. We have some videos online about them. And this, again, opened a lot of questions about reliability. So it's not about just doing the genome testing, but rather we want it to be reliable, regardless the environment that we are testing, because again, genomes is not things that special to human, but rather it exists in fish, for example, on the corpse, where we have like fruits, vegetables, viruses, bacteria, regardless if it is in the ocean, if it is on Earth, if it is in field, if it is even in outer space. So currently NASA is doing those tests in the space shuttle. Why is that? Because as you know, the space shuttle is a controlled environment. There is no hospital. Most of the time, there is no doctors on the shuttle itself. And if it's a disease happening there, regardless if they are taking it from the Earth, as they are sending some shuttles or some cargo to the space shuttle, they could bring some viruses from Earth to those places, right? Or this virus might already exist on the moon and we don't know, right? So it's really important to do those testing on regular basis so that they ensure that the astronauts in these space shuttle are safe and healthy. And if something wrong happening to them, they got sick or they whatever get worse over time, there is nobody to save them, right? They need to wait until they send someone from Earth to those places to do something. However, it's still very challenging. You know that the gravity in those places is almost zero. So it's really hard to pick those chemical molecules, put them in a machine where they will start floating on the surface. So it's really hard to control those chemical molecules, start breaking them down, do some reaction on them, and then try to sequence them and read those genomic material. One of the most famous problems that those people facing is the air bubble. So there are a lot of air bubble in the chemical molecule itself, causing problems on reading those genetic material. The other issue that we might face in genomics or bioinformatics, the things that Henrietta Lacks face, right? Which is reading your genomic material without your consent. And this genomic material could be correlated to your identity without even publishing the name or the owner of that sample. So how is that possible? If you're interested, read this paper about genomic privacy, where we show that is possible using some social media, some public information, public record from the government. You could correlate the ownership of that genomic material with some name. Although the hospitals sometimes do de-anonymization to the record, so they hide the name, they hide the birth, the date of birth, the place maybe, but sometimes they show the zip code. Sometimes they show some characteristics, especially the health record, like eye color, the hair color, and so on. And those now known location in the genome, you already can know that this location in the genome is responsible for the eye color. So you could read that from the genome that's published online, and then you could build the identity of the person gradually, one by one. So you know that now the eye color is blue. Okay, you can make a short list. And then the hair color is brown. Okay, that's another information. And then the zip code is this. Then you have now narrowed down the possibility of those person. And there is large number of studies who could figure out about 50% real identity of those 50% in one of the widely used and public database that accommodates a genomic material. And we study all of those cases in our paper called "Can You Really Anonymize the Donors of Genomic Data in Today's Digital World?" This is the paper. It's already online. That's why some DNA testing trying to do privacy-preserving DNA testing, which means that they destroy this record after they test you, and they try to hide your identity. They try to destroy the genetic material so nobody's using them later on. All of those are promises. We don't know if this is happening in reality, if they keep them or if they don't keep them. We don't know what is happening inside those companies. However, the applications are really limited by our imagination. It's not only health-related problems, but you can think about DNA storage, which is the thing we discussed last week. So this is the paper I mentioned in last week, published in Nature Biotechnology in 2020 in a collaborative work with ETH Zurich, where they demonstrate that you are able to store 200,000 terabytes of data in a single gram of synthetic DNA. So this is artificial DNA, still built out of the 4 molecules that we mentioned, and they mix it with some material like plastic, and then they could form shapes out of that as a toy or as a glasses and so on. And within those material, building the bunny, for example, the DNA is inside. So you need to take a swab out of this, or you melt down the bunny. You take the liquid, which has the synthetic chemical molecules inside it, and you start doing sequencing to retrieve the data. So you need to destroy, basically, those objects in order to retrieve the data. However, this is not the problem. Destroying is not a problem. You can destroy it, retrieve the data, and build the shape again. The most interesting part out of it is the long-term storage that you could have. So this bunny can last forever as long as you keep it in room temperature, for example, or safe environment where it won't get melted down, for example. Another application that's not related really to the health, but rather related to the marketing perspective. So this company in the UK tried to analyze your DNA. So they took sample from you, out of your consent, of course. So you send that sample to the company. They do the analysis for you. And then they will give you something like Apple Watch, where it records your DNA and some markers that correlate those DNA variation with some disease. For example, might tell you that you might have, for example, a certain disease, and that disease is not good to have that certain food. For example, you have allergy to strawberry, for example, right? So those information will be recorded in the Apple Watch, and whenever you go to the supermarket, you just scan the barcode on the product, and it will tell you if this is suitable for you or not, without reading the material, for example, or what is in those products. So those kind of things, like, you can think about how great you can build such application, and it's really limited by our imagination. There are many more applications, but one important thing to consider here when we talk about bioinformatics is not just building algorithm and makes a computer application out of it. It's not only that perspective, but rather we wanted to have what we call intelligent genomic analyses. And by intelligent, what we mean is that, can we make it real time? Think about critically ill child who's admitted to the hospital. Do we need to wait 3 days as what is happening today? We need to wait a few hours or a few days to get the analysis done, and then we start understanding what's happening with the child. So it would be really important and critical to get a real-time analysis for those children. And it's not only that. if we are able to analyze a single sample in 3 days, for example, how about 1,000 patients get admitted to that hospital at the same time? Do we need to wait 3 days for each of those patients in a row, such that we get like 2 months to finish those analyses? How many analyses can we do in parallel, right? So that is what we call scalability. And we also think about the energy efficiency, portability of those analyses. We don't want a huge machine to be set in the wet lab and avoiding us from doing the analysis next to the patient. Think about the blood test now for many of the disease that we could do in place right away next to the patient. We just take a blood drop, we could do the analysis right away and so on. We would like to have something like that with the genomic material. But also about the energy efficiency. For example, now most of the companies try to build AI center with nuclear energy, for example, because the amount of energy that requires. So if we are going to encourage people to do genomic analyses for the entire population, probably that is also a solution to build those specialized center for genomics that get fed with energy using nuclear plants or whatever we call them. And this is something not efficient, is not feasible to be done in the entire world. So we are looking for really energy efficient solution or hardware to do those analyses. Another important aspect is the privacy, and something more important, even the accuracy, as what happened in New York subway. That could raise an important alarm about the false positive of those analyses. We don't want to tell a person that you might have this disease and then they start treatment based on that, but the testing was wrong. That's why this field is really interdisciplinary. There are a lot of things working together. That's why we need people from all fields, from mechanical engineering, electrical engineering, chemical engineering, computer engineering, math, biology, chemistry, and so on. If you are really interested in knowing more about what we covered today, I would recommend you reading this. This is a very long paper because we address a lot of things around this area or topics that we discussed today. So I would recommend everyone to read it whenever you have time. You can skip some of these sections if you are not interested. But one of the most important sections that we will discuss in the next lecture, which is sequencing. So if you are interested in that, you can start reading about sequencing from this paper. And you can read also why until today we are not able to read the entire genome as a single sequence. And we need to break it down and read each of those pieces individually. All right, that was it for today. Hopefully, you learned something new. Hopefully it was interesting to you. Next lecture, we'll get more hands-on examples about sequencing. We are going to see the data in real, especially those pieces that we got from the sequencing machine. We still have about 10 minutes or so. So if you would like to discuss, ask some questions, anything not clear, or maybe you have some real story to share with us about bioinformatics. All right, hopefully that is a good sign, means everything was well and smooth. You understand the content of the course, or at least this lecture. All right, it was nice seeing all of you. Hopefully next week the weather will be better and then we will meet in person. Please stay safe and warm and see you next week. Thank you so much.
Thank you, Professor.
Thank you, Professor.
Thank you.
Thank you, Professor.
You're welcome. Have an amazing day. Bye-bye.
Bye, Professor.